Good morning. Nice to meet everyone. My name is Heather Stevenson Lerner. I'm a professor in the Department of Pathology at the University of Texas Medical Branch in Galveston, Texas. And I'm a physician scientist that studies the liver microenvironment and different types of chronic liver disease. And I've been using Visiopharm software now for almost 7 years, and we've had a lot of great data generated with the projects we're doing, and I'm really excited to tell you more about those today.
Cool, exciting. So let's start with the first question we have for you. Can you describe the primary focus of your research and the specific pathologies you are studying with Visiopharm software?
Yes, so I've been the only liver pathologist here since 2014. So for over 10 years, I've signed out every single liver biopsy. So the great thing about Visiopharm and the tools that we use to study the hepatic microenvironment is that we can do the work on formalin-fixed paraffin-embedded tissue. And as a pathologist, I have access to over 30 years worth of this archive tissue. I particularly am interested in liver tissue, but we have every tissue that basically that comes out of a patient here that we save, which I would highly encourage everybody to save your tissue, because now there's so many great platforms that we can do on this tissue. And the cool thing about this is when I go back to these archived liver tissue in different types of chronic liver disease, for example, steatotic or fatty liver disease, viral hepatitis C, immune-mediated liver diseases such as autoimmune hepatitis or primary biliary cholangitis. And even more interestingly, we have access to samples from infectious diseases like Ebola virus here at UTMB. We're known for emerging infections. So we've been able, because these archived tissues have been deactivated, we can work with all these infections we normally would have to be in BSL-3 or BSL-4 laboratories with. So that's been great. But the primary focus of our research is to study the hepatic microenvironment. We're very interested in the macrophage-mediated immune response, because it's very important in activating a pro-inflammatory microenvironment and causing scarring or fibrosis in the liver. And so what we do is we use these archived liver biopsies to stain different types of antibodies. We look at gene data, but also where Visiopharm comes in is we're looking at several different antibody markers, our multiplex staining for all these different types of diseases. For example, we do a 6 different antibody marker for macrophages, and Visiopharm has been really, really integral for helping with our success, because a lot of these markers we're looking at are in the same compartment on the same cell, and there's just no other software platforms. Over the years, we've tried a lot of different things, and we found that Visiopharm is the best at actually helping us differentiate these different phenotypes within the hepatic microenvironment in these different diseases.
Cool. And what challenges or limitations in your research led you to consider using AI solutions like Visiopharm, for example?
Well, I work a lot. When we first started doing this work, I still am collaborating with MD Anderson Cancer Center, which is about an hour up the road in Houston, Texas. And I work with Dr. Jared Burks, who runs their lab that does all of their spatial approaches there. And so a lot of these platforms that we're using now obviously came first in the tumor microenvironment. So for example, you study tumor and non-tumor. And this is really helping us with our precision medicine approaches. So I was like, well, if they're doing this up the road in the cancer microenvironment, here at UTMB, we specialize in inflammatory or medical liver diseases and infectious diseases. So why can't I do the same thing that they're doing in tumors in non-tumor tissue? We were one of the first ones doing it here at UTMB, and now it's really caught on. Obviously, the spatial field has grown so much in the last few years. But Dr. Burks and I both came to the conclusion that Visiopharm was the best. At that time, we were trying a bunch of different things. I mean, we tried Definiens, we tried Halo, we tried everything. They all have their own, even ImageJ and things that are available online, and they all have their pluses and minuses. But as far as what we have found, we both settled on using Visiopharm. I believe they have an institutional license there, and we have a license here at UTMB. We've just had the best luck. The AI feature for your cell segmentation, which if you don't get your cell segmentation right at the very initial step, everything downstream is going to be off. So that AI-driven cell segmentation has been crucial for us, because all of our downstream applications and algorithms after that moving forward, if that's accurate, your data is going to be more precise and reproducible. In addition, when we first started using Visiopharm, we wanted to make sure that the data was, again, reproducible patients are different. We got to make sure what we're seeing was accurate. So what we did was we actually collaborated with the bioinformatics group at the University of Michigan, which are one of the best in the country. And so what we did was we took our multi-component TIFF files from our experiments. As you know, that's the main files we use for Visiopharm. And then we also took our numerical data from Inform software from like a Vectra or Polaris. And for those, it's cell seg files, which are numerical kind of set on thresholding. And so we would send the same experiments from the same patients, but just two different types of data analysis, one being sent to Michigan, and then the one we did ourselves on Visiopharm to make sure the results we had were the same. And we did come up with the same. All of the work I'm going to be showing you today, we had the same results by looking at the data in two completely different ways. But the nice thing about Visiopharm is we now have it in our own laboratory, so we don't have to rely on anyone else for the analysis. So it's been great to learn how to use it over the years.
Perfect. I think you answered a little bit of the question number 3, but I'm going to ask again. So how has the use of Visiopharm software improved the quality and accuracy of your research outcomes?
So we really, as we know, we're really trying to look at the spatial or keep the hepatic or liver architecture intact. So there's a lot of great things out there. I mean, when I was a graduate student, I did flow cytometry in the liver. But for flow cytometry, you have to homogenize the entire liver tissue, right? You need fresh tissue, number one, not formalin fixed. That can be a challenge, especially with human patients. And then you're homogenizing the liver and the cells and separating from, you don't really know. You can tell how many of what cell type's there, but you can't tell where it is. For example, is it in the portal tract of the liver? Is it in the lobule of the liver? Is it next to, what type of cells are next to each other? Who's talking to who? Or neighbor analysis. You can't do that with flow cytometry. In addition, in single cell RNA-seq, you're kind of recreating the liver microenvironment, but you're still not really preserving the architecture. Visiopharm allows you to analyze these different cell types. You can look at the phenotypes. You can look at the level of expression of different markers. You can do TSNEs or UMAPs to determine the relationship of cells to each other. So just the power of what you can do by preserving the liver architecture is something Visiopharm allows you to do that these other applications don't. In addition, when you're looking for something with a very small signal, for example, we also use nanostring. We homogenize the liver and we look at whole RNA signatures using nanostring, which can be powerful. But what happens with nanostring is if you're looking for one cell type with a druggable target or expression of a single marker, sometimes that's diluted. Right? So by homogenizing the liver tissue, I'm going to miss a signal. But if I'm able to go into the liver and actually look at the macrophage, go segment on CD68 positive macrophages or do tissue segmentation and actually cluster those macrophages, then look at the signal, I'm going to see things that I might miss by homogenizing the entire liver. So that's something that Visiopharm is very powerful for doing.
Cool. And in what ways did Visiopharm software prove to be the only viable solution for your research needs compared to other tools or methods you considered?
Well, I think really there's several important things. As I mentioned already, but is the being able to stain the multiple antibodies on the same compartment of the cell. We do try to combine, different antibodies. Like we have some really nice antibodies that are nuclear, cytoplasmic, and membranous, which that does help. But we do have several antibodies like CD68, CD163, CD14, and CD16, which are in the same compartment, just different fluorophores or opal fluorophores that we use. So that Visiopharm did the best at being able to actually separate cells with antibodies in the same compartment, number one. Number two is you can use a lot of the algorithms straight off their website. So that's helpful, or your website. But we also, the nice thing we've been able to do is once we develop our algorithms, we can actually plug those algorithms into different chronic liver diseases with very little tweaking. So once we save our customized algorithm, we've been able, for example, to make an algorithm for chronic hepatitis C or HCV, and then plug, by changing just a few of the applications or just tweaking it a little bit, plug in patients with autoimmune hepatitis. So we have those algorithms. Once we have them saved, we do a little modifications and we can do the same thing with other diseases, particularly if you're looking at like the same macrophage antibody panel. That's very helpful. The other thing is the training sessions. that we've been able to use from Visiopharm. Thank you very much, Justin. We've been meeting with him for several years now. It's part of our software package. I wouldn't say we we go through times where we're using Visiopharm a lot, and we have weekly meetings with him, or if the software update comes or there's a particular question, we meet more frequently. But those training sessions have been crucial to our success. And also the customer support whenever we have a question, and always being available to help us answer our questions so that we're not being held up, has been great. So I would say that those are the main things that have been really helpful.